Hi, I'm Adria LeBoeuf, and today I'm going to tell you about some biomarkers of aging that we find in a socially exchanged fluid and that I think underlie a form of distributed metabolism in social insects. So much of biology can be thought of as a series of trade-offs about resource management. Trade-offs that, when differentially managed, can lead to the incredible diversity of species that we observe around us. The evolution of aging over the lifetime of an individual can be viewed as a trade-off about how much to invest in somatic maintenance versus reproduction, essentially the longevity-fecundity trade-off. Another classic aging trade-off occurs within an individual, between the germline and the soma. Disposable soma theory suggests that metabolically expensive germline is maintained at the cost of an aging soma. But how does this work mechanistically? Can costs and benefits flow between tissues? So through studies that can actually ablate the germline, we can see that somatic tissue can repair more quickly in the absence of germline. And this suggests that there is indeed a flow of both costs and benefits between the two in the form of metabolic signals and cues. Social insects provide an excellent window into the biology of aging. First, on an individual level, they seem to subvert this classic longevity-fecundity trade-off with long-lived, like 30 years, highly fecund queens and short-lived, like 1 year, sterile workers. Second, they provide a third scale over which we can view these trade-offs about resource distribution, the colony scale. So to understand this, I need to tell you a bit more about the unique biology of ants. While each individual ant you see crawling over your kitchen counter is a multicellular organism in and of herself, the unit of reproduction in most cases is the colony, not the individual. So here is the life cycle of a stereotyped ant species. A young queen emerges from her natal nest for the first time. She mates once and uses the stored sperm from one mating for decades, laying millions of eggs. These eggs, if fertilized by those stored sperm, can be epigenetically guided towards either a queen fate or a worker fate, akin to germline and soma. So the workers fall into two somatic tissue types, nurses and foragers. In a worker's approximately 1 year long life, she starts as a nurse, caring for young larvae, and eventually she shifts towards foraging outside the nest. So all of these individuals, aside from the queen, are sterile workers, whose only fitness is indirect fitness, achieved when the colony as a whole finally produces new sexuals. So this is why such colonies can be considered superorganisms. They only reproduce at the colony scale. So thinking about aging at the scale of the colony, we can see reflections of both of these trade-offs, these classic trade-offs, the germline soma trade-off between investment in queen tissue or worker tissue, and then the lifespan trade-off, where in early colony life, the colony must invest its resources in rapid growth, and then only once it's mature, it can shift to reproduction and dispersal in the form of sexuals. So from gene expression studies in social insects, we know that some of the underlying molecular mechanisms of this transition are deeply linked to nutrient sensing and metabolism. But because metabolism is fundamentally a very individual accounting problem of what comes in and what comes out, it leaves many open questions in a system that's distributed across thousands of individuals. So how does the queen avoid aging herself? How are those metabolic costs distributed between individuals? And how does the colony collectively go through this ant colony puberty, transitioning from producing workers to producing queens, and then maintaining that balance? So I propose a social fluid exchange called trophallaxis. The social transfer of bodily fluids tends to only occur in behaviors that straddle the border between cooperation and conflict, things like mating and parental care. And social fluid exchange itself represents a kind of private communication channel that can be used to exchange bioactive materials. Consequently, socially exchanged fluids can be hotbeds of manipulation and adaptive evolution. So today I'm going to explain how I think that this behavior gives us a window into superorganismal physiology and into these fundamental evolutionary trade-offs. So trophallaxis is the ingestion by one individual of a fluid that's been excreted, secreted, or regurgitated by another. The most well-known form is a mouth-to-mouth exchange that occurs between adults that spreads both exogenously sourced food and endogenously produced material throughout the colony. And from my previous work, we know that trophallactic fluid contains endogenously produced proteins, small RNAs, hydrocarbons, hormones, and other small molecules. So for many social insect species, where trophallaxis is the primary means of food sharing, every individual in the colony is connected through this network of fluid exchange. And so we can view trophallactic fluid as a kind of social circulatory system that distributes nutrients and signals to each member of the colony. So if this is indeed anything like a social circulatory system, we should see evidence of our trade-offs within components transmitted by trophallaxis. So to look at this, we analyzed the proteins in trophallactic fluid samples from single ants and from colonies at different stages in their life cycles, aiming to leverage these conflicts at different scales. So my postdoc, Sanya Hakala, collected trophallacic fluid from individual nurses and foragers from lab colonies of Camponotus floridanus, expecting to see differences relating to individual lifespan and different somatic tissues. At the colony scale, we wanted to access the maximum dynamic range for different priorities regarding resource use. So we expect young colonies to prioritize rapid growth and mature colonies to invest in new queens and males for dispersal. Lab colonies of this species, though, rarely grow to the thriving maturity that they do in the field. So we went to the field to do this, the Florida Keys. And we also took this as an opportunity to see how trophallactic fluid changes when colonies move from the field to the lab. Lastly, we looked at whether we could see any differences across our population over islands. If this is fundamental physiology, we shouldn't really see any differences there. Now, to get at the molecular machinery underlying these trade-offs, Sanya and I used 3 different statistical methods and overlaid the results, retaining only proteins significant in at least 2 of our 3 analysis methods. The approaches were actually very complementary, and combining them allowed us to really separate signal from noise. Notably, in our sort of negative control comparing between islands, we found no robust differences. So here are significant proteins where color indicates fold change, and yellow dots indicate proteins significant in 2 out of 3 methods. A few salient results. So comparing young and mature colonies, 2 things jumped out. Some of the top hits for mature colonies were large nutrient storage proteins, things like hexamerins. So in insect development, these proteins are the means for larvae to accumulate resources and then repurpose them during metamorphosis through a sort of organism-level form of autophagy. Finding these proteins being socially transmitted is very neat because it puts forward a mechanism for how to sort of nutritionally bring about new adult morphologies through consolidation and investment of resources across individuals. Young colonies, on the other hand, had significantly more abundant simple sugar processing enzymes, things like amylase and glucose dehydrogenase, suggesting that they use their nutrients as soon as they get them. So we can view these different hits at different points in the colony life cycle as different strategies: resource consolidation and investment for rich, mature colonies, and spend your whole paycheck on payday for the young colonies. For our nurse versus forager comparison, again, we see these fast sugar processors, this time significantly high in our foragers. In nurses, a number of their hits are in anti-aging proteins and proteins that are protective against metabolic stress. So generally, one can produce antioxidants for 2 reasons. Either you suffer from metabolic stress, from oxidative stress, or you want to suffer less. And so we have a few hypotheses for what these could be up to. They could be about maintaining nurse individual lifespan. They want to suffer less. Nurses are nearest to the queen in the colony's social network, so these could be fed to the queen to protect the germline. They want the queen to suffer less. Or these proteins could reveal the metabolic costs of larval rearing, which is done primarily by the nurses. The nurses are suffering. So fundamentally, there are more experiments to do here. So as you can see, this social fluid exchange is a keyhole through which we can observe evolutionary trade-offs and distributed metabolism at play in superorganismal physiology. And going beyond social insects, this system may help us better understand how multicellular organisms and their tissues communicate and share resources and costs cooperatively. So I want to give a big thanks to my funding sources and to my lab, especially Sanya. Thank you.